In this video, I'm going to be covering the entire Module 5 for GCSE Biology. This video covers all of the content that is applicable to both combined science and triple science students. And as always, there are some free flashcards that you can access to help you test your knowledge on Module 5. Just click the link in the description, fill in your details, and we'll send them over to you immediately. Also, in the link in the description, you'll find a link to my resources where you can access free revision resources on the whole of GCSE Biology, including Module 5, which is probably one of the trickiest topics, I would say. So click the link and check out my resources now if you haven't done so already. Okay, let's get started with this video. So we're going to be doing Module 5 in this video. This is homeostasis and response. We're going to cover in this video all of the content that is applicable to both combined science and triple science, but do make sure you're subscribed because we're going to be posting another video very soon on the triple science only content because there's absolutely loads of it in Module 5. Let's start with what is homeostasis. So homeostasis is the regulation of internal conditions of a cell or an organism, and it's needed to maintain optimum conditions. For example, optimum conditions for enzyme action and cell function. So we're basically trying to maintain a stable internal environment to make sure the conditions are optimum for our cells and our enzymes. Now, homeostasis is maintained using what we call negative feedback mechanisms. So let's just use a diagram to help you understand what that is. Obviously, we want to keep the conditions at normal levels. So for example, our blood glucose or our temperature, we want to maintain it at a normal level. So if there's any increase in that level, in that temperature or that glucose concentration, the increase will be detected. That will mean that a response is coordinated to bring the level back down to normal. And equally, if there's any decrease in the normal level, like our temperature drops below 37 degrees, the decrease will be detected. A response will be coordinated to increase the level back to normal. So we're maintaining that stable internal environment to maintain optimum conditions for enzymes and our cell functions. Now, negative feedback uses automatic control systems. Now, this could involve nervous responses with our nervous system and chemical responses, which involves hormones, but all control systems will include the following things. So there are cells called receptors, which will detect a stimulus. So any change in the environment will be detected by our receptor cells. There's coordination centers. Now, this will be your brain or your spinal cord, which we call our central nervous system. They will receive and process the information from the receptors about the change in the environment or the stimulus. There are effectors, which can either be muscles or glands, which will bring about the response. Muscles will contract, glands will secrete a chemical substance such as a hormone, but we'll go through all of this in more detail. Remember, examples, because I haven't got it on the slides, but examples you will look at will be blood glucose regulation. If you're triple science, you'll also do temperature regulation, and you'll also do regulation of water content in your blood or osmoregulation. The next thing we're going to talk about is a reflex action. So what is a reflex action? This could be a 2-mark question on your exam paper. A reflex action is an automatic and rapid response to a stimulus, so to a change in your environment. And why do we have them? Well, it's to help protect our body tissues from any damage. So for example, to prevent burning if we touch something that's really hot, or to prevent damage to our retina if the light intensity is too high. But it's really important to remember that reflexes do not involve the conscious part of the brain. So you don't have to think about them. And this is why they are so fast. And this is why we say they are automatic. So let's have a look at an example of a reflex action. It's shown in a diagram like this that we call a reflex arc. So we've got receptors that will detect the stimulus. Now, in this example, they're temperature receptors or thermoreceptors in our skin, in our fingers. That stimulus will be detected and that will trigger an electrical impulse. We say electrical impulse, everybody. We don't say message, we don't say information, we call it an electrical impulse. And that's going to travel from the receptor down a sensory neuron. And the sensory neuron carries the electrical impulse into our central nervous system, usually into our spinal cord. Unless maybe the reflex action involves your eye, then it would carry, the sensory neuron would carry the electrical impulse into your brain. But anything kind of below the head, the sensory neuron is going to carry the electrical impulse into the spinal cord, which is part of your central nervous system. Remember, your CNS is your brain and your spinal cord. Now, from there, it's going to travel down a relay neuron, which is in the central nervous system. So it's in between the sensory neuron and the motor neuron. Here it's in the spinal cord. Then the electrical impulse is going to travel back out down a motor neuron. And the motor neuron carries the electrical impulse to what we call the effector, which will cause the response. Now, effectors can only be muscles or glands. Here, the effector is a muscle because the response is going to be that the muscle contracts to move your hand away from that hot object, whatever was increasing the temperature, basically to protect your skin from any damage. Let's go through all of these key parts then of a reflex action. So you've got some nice definitions. So the stimulus is a change in the environment that you need to respond to. So it might be an increase in temperature, it might be an increase in light intensity. The receptor is a cell that detects the stimulus. So for example, temperature receptors in the skin or light receptors, photoreceptors in your retina, they're going to detect the stimulus. And I'm just going to add on here, they convert the stimulus into an electrical impulse. So if it asks you what is a receptor and it's 2 marks, you can add that second bit on. Then we've got our sensory neuron. So the electrical impulse travels down a sensory neuron, and that carries the electrical impulse from the receptor into your central nervous system, usually into the spinal cord. If it does go to the brain, say it's a reflex action involving your eye and the receptors in your retina, then the sensory neuron would carry it to the brain, but not the conscious part of the brain, okay? Because reflex actions are really fast, automatic responses that do not involve the conscious part of the brain, or you don't have to think about them. Okay, so the sensory neuron is carrying the electrical impulse from the receptors into the CNS. In the CNS, there'll be a relay neuron, which is a short neuron. The electrical impulse will travel down the relay neuron, and that connects to the next neuron, which is the motor neuron, and that carries the electrical impulse to the effector. And the effector, dead easy, it can only be one of 2 things. It's going to either be a muscle or a gland, and they will cause a response. Now, if you're asked about the responses, you say muscles contract, don't say they move. Muscles contract, which will cause movement. Or if the effector is a gland, it will secrete a chemical substance, which is usually a hormone. So that's the order, and I suggest you memorize the order of structures in a reflex action. So your stimulus is the change in the environment detected by the receptor, electrical impulse down the sensory neuron, then the relay neuron, then the motor neuron to the effector, which will cause the response. Now, the response, if you're talking about a muscle, you must say the muscle contracts, and then you can say to move the hand away from the hot pan. Or the other example we've been talking about is the eye. The muscles in the iris can contract to reduce the size of your pupil, for example. Let's see if we can label all of these things on a reflex arc diagram. So let's start here, because D would be the receptors. Yeah, so these are cells that detect the stimulus. I think the stimulus here is going to be heat or an increase in temperature because it looks like they're touching a hot pan. So the stimulus is the change in the environment, so the increase in temperature because they've touched the hot pan. It's detected by receptors. They detect the stimulus. These would be found in the skin. These are obviously temperature receptors because they're going to detect that increase in temperature. Remember, they convert that into an electrical impulse, and the electrical impulse is going to travel down our first neuron, which is our sensory neuron, which carries the electrical impulse from the receptor all the way in to our central nervous system. This is obviously our spinal cord, and in the spinal cord, in the central nervous system, there will be a relay neuron, which is a short neuron that connects the sensory neuron to the motor neuron. So the impulse is going down the relay neuron, then it's going along G, which is the motor neuron, and the motor neuron is carrying that to F. So F is the effector. In this case, it's a muscle, which means the response will be the muscle contracts, don't forget to say contracts, to move the hand away from the hot pan. Just one more thing to mention here, another word that you might hear. The spinal cord in this example is acting as what we call the coordinator in this reflex action because it's receiving the electrical impulse along the sensory neuron. It's going to coordinate the response and send the electrical impulse out down the motor neuron so that the muscle will respond appropriately. It will contract to move the hand. So the spinal cord is referred to as the coordinator. If this reflex action did involve the brain, then the brain would be the coordinator. Basically, the central nervous system we describe as the coordinator. Now, something we've not mentioned yet, there are synapses between neurons. Now, you need to know what a synapse is. So it's a gap or a junction between two neurons. So, for example, between the relay neuron and the motor neuron, there'll be a gap. Or between the sensory neuron and the relay neuron, there'll be a gap. There'll be a synapse. Now, the problem is electrical impulses cannot cross these synapses. They cannot cross these gaps, just the same way in physics. If you make a circuit and there's a break in the circuit, the electricity cannot cross. So how does the information pass across a gap, across a synapse? Well, what happens is when the electrical impulse reaches the end of a neuron, it triggers the release of chemicals called neurotransmitters. But at GCSE, you can just say it triggers the release of chemicals, okay? So it's not an electrical impulse that's going to cross the synapse. It's a chemical. And the chemicals will diffuse across the synapse, bind to the next neuron, which will trigger another electrical impulse. Now, because transmission across a synapse involves diffusion of chemicals, diffusion of a chemical is slower than the transmission of an electrical impulse is. So synapses do slow the reflex action down slightly, but they're still super, super fast, right? We're talking milliseconds here. But they do slow it down slightly because you've got your really quick electrical impulse in the sensory neuron, then the diffusion from a high concentration to a low concentration of a chemical across the synapse, then an electrical impulse along the relay neuron, then the diffusion of the chemical across the next synapse, and then the electrical impulse is transmitted along the motor neuron. So that's how it works. Synapses are found between neurons. They are junctions between neurons. So as I've said, you will get one between the sensory and the relay neuron and one between the relay and the motor neuron. There'll also be another one between the motor neuron and the effector, which could be a muscle. We'd actually call that synapse a neuromuscular junction, but you'll be learning more about those at A level. For GCSE, it's really just remembering that chemicals have to diffuse across a synapse because the electrical impulse cannot cross the gaps between the neurons. Right, there is a required practical linked to this topic of the nervous system. It's all about reaction time. So let's talk about that. Reaction time is the time you take to respond to a stimulus. Now, typical human reaction times vary slightly, but they're pretty fast, between 0.2 to 0.3 seconds. Now, there are factors that will affect your reaction time, which is why we do this practical, because we can investigate how one of these factors affects reaction time. So, for example, whether or not you've had caffeine, because caffeine is a stimulant. Age might affect reaction time. Does it slow down as you get older? If you're tired, does that slow down your reaction time? If you've practiced or you've trained to do something over and over again, do you get faster at it? If there's any distractions in the room, would that slow down your reaction time? Like if there's loud music or someone's talking to you. If you've had any drugs, medications, or you've been drinking alcohol, that would likely affect your reaction time. So your required practical is to investigate how one of these factors would affect the reaction time. For example, you might have looked at caffeine, drinking coffee or cola versus not drinking coffee or cola. You might have looked at distractions in your school classroom to see if that affects your reaction time. Now, we're going to look at an example method. I'm not saying this is the exact one they'll use on the exam, but it will probably be something very, very similar. We're going to look at the effect of caffeine on reaction time, and we're going to look at an example method. So we would use the ruler drop test for this, because it's a really simple way to investigate reaction time. You don't need a computer or any specialist equipment. You literally just need a ruler. So your test subject, so your partner in class, would sit with their arm resting on a table. You would then hold the ruler vertically just above the subject's thumb or forefingers, like you can see here. And you'd make sure you had the 0 centimetre mark down at the end, just above their thumb and their forefinger. You drop the ruler without warning, because you do want to test their reaction time. And you would record where the ruler is caught. So, for example, they might catch it at 15 centimetres. Now, obviously, with any investigation, we always say you have to have repeats. So you'd make sure you've got at least 3 repeats, so you could calculate a mean distance where the ruler was caught. So they might catch it at 15 centimetres, 16 centimetres and 17 centimetres. So you could divide that by 3 and work out that the mean distance the ruler was caught at was 16 centimetres. Now, you might be thinking, that just gives me a distance. How do I actually know their reaction time? Well, you can look up the reaction time that corresponds to where they caught the ruler. You can actually find this in a table online. So your teachers might have given you this table, but you can look up, on average, they caught the ruler at 16 centimetres. That would equate to a reaction time in seconds or milliseconds, which you could then look up online. Then you'd have to repeat the whole investigation, but you'd have to give the subject a caffeinated drink. So e.g., you could make them drink, I don't know, I was going to say a litre of cola. That's probably too much. Let's say 300 mils of cola or something that is caffeinated, maybe coffee. Wait 15 minutes, obviously have the time for that caffeine, that stimulant to take effect, and then you would repeat the whole investigation. So you do the same ruler drop test, controlling all necessary variables. And again, you'd repeat it another 3 times, making sure you get the mean distance where they caught the ruler and figuring out what reaction time that equates to by looking it up online. Now, should you just do this with one participant? No. Ideally, the more people you do this on, the better. And this is true for any investigation. The more repeats, the better. So any investigation you're planning when you're using people, participants, you would talk about doing this on a large number of people. You could do it on every single person in your class. I know that would take a long time. But in your exam, you would say, repeat this on a large number of people so you can get the mean reaction time before drinking the caffeine and the mean reaction time after drinking the caffeine, because the more data you have, the more reliable your conclusions are going to be. So you'll be able to make a comparison. Is there a difference between their mean reaction time before drinking the caffeine and their mean reaction time after drinking the caffeine? Now, what's really important in any investigation is that you are able to identify your independent, your dependent, and your control variables. And if you're planning a practical, I always recommend to my students that you put this into your plan so it's nice and clear to the examiner. So our independent variable is the one we change. Yeah, I for independent, I change it on purpose to see if it affects the results. So our independent variable was whether or not the participant had consumed caffeine. So caffeine versus no caffeine. It might be distractions versus no distractions. It might be 50-year-olds versus 20-year-olds, whatever variable you are investigating, whichever variable you are changing. So in an exam, if I was planning an investigation, I would make a point of saying my independent variable is whether or not the participant has consumed caffeine. So I would do this investigation before drinking caffeine and after drinking caffeine. Now, the dependent variable is the one you measure. Yeah, the one you're going to measure, the one you're going to record in your table when you're filling your table in class. Now, in this case, it's reaction time, or you could say it's the distance at which they caught the ruler. You could say either, because you're measuring the distance where they caught the ruler, but you are going to use that to get the reaction time. So that's your dependent variable. And in an exam, again, I would say my dependent variable is the reaction time. I'm going to find this by measuring where they caught the ruler and then looking up the reaction time online. Control variables are so important. And if ever you're planning an investigation, I always tell my students to include at least 3 so you can access the top tier of the mark scheme. So here are some examples of control variables that you would have to keep the same, because if you do not keep these the same, they could affect the reaction time and basically make your results invalid. So we control variables to get valid results. Don't just say to make it a fair test. That is not good enough. We control them because they would affect our dependent variable. They would affect a person's reaction time, and that would mean the results and our conclusions would be invalid. So things you could give, you'd have to drop the ruler from the same height each time. So the same height above the participant's hand. Do it at the same time of day maybe, so the person is not tired. Don't do it at 10 a.m. versus 10 p.m, for example. Use the same dominant hand. So if they're right-handed, they catch it with their right hand, or if they're left-handed, they catch it with their left hand. Because if they use different hands, that might affect how quickly they catch the ruler. Make sure you have same conditions in the room you're doing your investigation, same lighting, same noise, same level of distractions, i.e. no distractions ideally. Same volume of cola or coffee for all participants. And this one's quite important, the same time left after drinking the cola before doing the test. You can't get them to drink the cola and then do the test immediately, because it won't have been absorbed into their bloodstream and it will definitely not affect their reaction times. So make sure you highlight at least 3 control variables if planning this practical, and to be honest, for any practical. I always tell my students to think about IV, DV, CV repeats. So what are you going to change and how? What are you going to measure and how? 3 things you're going to keep the same or control, and make sure you have at least 3 repeats. But obviously, if you're using people, you want as many people as possible, and maybe repeat 3 times with each person, so you've got plenty of data to make sure it's reliable. Let's move away from the nervous system now and start to think about the endocrine system. Now, the endocrine system, forget about nerves and electrical impulses. This is a system of glands that secrete hormones. Now, you might be asked, what is a hormone? What is the endocrine system? So the endocrine system includes all of the glands and all of the hormones that they secrete. Now, hormones travel through the bloodstream to target organs, because these target organs will have receptors for the hormones. So hormones don't affect every single organ in your body. They will only affect the target organs, the ones that have the receptors for that hormone. They travel in the bloodstream, and hormones are described as chemical messengers. Most hormones, not all, but most hormones are proteins. Some are steroid hormones, so some are made of lipids, but most hormones are proteins. They all travel through the bloodstream to target organs, and we describe them all as chemical messengers. So lots of important key things to say there. Let's make sure we can label the key endocrine glands. So we've got the pituitary gland, which is in the brain. Now this secretes hormones such as FSH, LH, and ADH. Now I'm not going to go through all of those on this slide because they're going to come up later. For example, FSH and LH are hormones involved in the menstrual cycle. So we'll cover them when we do the menstrual cycle. And ADH is a hormone involved in controlling the water content of the blood. So we'll do that in our triple science video when we talk about the kidney and osmoregulation. But for this part of the spec, you just need to be able to name the glands and give examples of the hormones they secrete. The thyroid gland, which is in the neck, this one secretes thyroxine. The adrenal gland, easy to remember, adrenaline. Now you have two adrenal glands, one above each of your kidneys. The pancreas, again, we'll go through these hormones later because they come up in blood glucose regulation. They secrete two hormones that control your blood glucose, insulin and glucagon. Now females have ovaries and they secrete estrogen and progesterone. So we'll cover those when we do the menstrual cycle. Males don't have ovaries, they have testicles or testes, and they secrete testosterone, which is a male sex hormone. I've just remembered another one for the pituitary gland actually, thyroid stimulating hormone, which we're going to be covering again later in this video because thyroid stimulating hormone stimulates the thyroid gland to secrete thyroxine. Now, the pituitary gland is often known as the master gland because these hormones that it secretes control other glands in your endocrine system. So you might hear the pituitary gland described as the master gland. Now, some of the questions that come up on this do ask you to compare control using your nervous system with control using your endocrine system or your hormonal system. It's probably only going to be 3 marks, maximum 4, but we'll go through some of the comparisons or some of the differences that you could be giving. So the endocrine system uses chemicals or hormones, which are chemical messengers, whereas the nervous system is using electrical impulses. Hormones travel in the bloodstream, whereas electrical impulses travel along nerve cells or neurons, like the sensory, the relay, and the motor neuron. Now, the endocrine system, because it uses hormones and because they have to travel in the blood, it is slower acting, whereas the nervous system uses electrical impulses, so it's very, very fast responses. We're talking millisecond responses. Endocrine system, the responses do last for longer because the hormones will remain in the bloodstream for longer, whereas the nervous system responses are very short-lived. Endocrine system, the hormones target specific organs or target organs that have the receptors for the hormone on their surface, whereas the nervous system targets effectors, so glands or muscles. Examples of responses, well, the endocrine system, we're going to look at blood sugar control, blood glucose control. Nervous system, we've looked at reflex actions, which involve muscle contraction. I mean, we've probably got enough by now, but the endocrine system does have more general effects because there can be multiple target organs. So one hormone can affect multiple organs, whereas the nervous system, it's very localized, it's targeted. It will affect a muscle or a gland. The endocrine system obviously involves glands, such as the ones we've just labelled, including the pancreas, the pituitary gland, the thyroid gland, whereas the nervous system involves your brain and spinal cord, which are called your central nervous system, and then all of your other nerves, sensory neurons and motor neurons that you find throughout your body. Now, this is a lot of information on one slide, so I would probably just try to remember maybe the first four. They're probably the easiest. And if you do get a question asking to compare, make sure your responses are comparative. For example, the endocrine system response is slower, whereas the nervous system response is much faster. So the difference is really, really clear. The word whereas is good for these as well. So like the endocrine system uses hormones, which are chemicals, whereas the nervous system uses electrical impulses. Okay, so we're going to look now at an example of homeostasis, maintaining our blood glucose concentration. This is an example of homeostasis. It uses negative feedback, and it's also an example of control via the endocrine system because it uses hormones. So it's a really important one to learn, and it comes up a lot. Let's start by thinking, why is it important that we control our blood glucose? Why do we want to keep it at a normal level without massive peaks or troughs? Well, glucose, which is a simple sugar, is needed by all your cells for respiration to release energy. So we've got to have a supply of it at all times. But if you have too much glucose in your blood, which would be called hyperglycemia, don't have to know that word, but high blood glucose, water would leave the cells by osmosis and your body cells would start to shrivel and shrink. Because if your blood plasma is full of glucose, that's a very concentrated solution. Your cell solutions would be more dilute. So water would start to leave your cells. Equally, if you have too little glucose or low glucose, hypoglycemia, then there'd be not enough glucose for respiration and therefore not enough energy released for your cells. So it's really important that we maintain a constant blood glucose level. And we do this with negative feedback, which means if it rises, there's a response coordinated to bring it back down to normal. If it falls, there's a response coordinated to bring it back up to normal. And this response involves hormones. So let's look initially at when your blood glucose level rises. Now, this would be after eating carbohydrates. If you eat starch, it's digested into sugars, or if you eat sugary foods, sweets, cakes, whatever. Even fruit has a lot of sugar in it, right? So after eating carbohydrates, glucose is absorbed into the blood, and that happens in your small intestines, which you should know from your paper 1 revision. Now, the pancreas detects the rise. So that's acting as the receptor cells, right? It's detecting the change in environment. It's detecting the rise. And the pancreas is a gland, so it releases the hormone insulin. Okay, so when blood glucose rises, it's insulin that is secreted from the pancreas. What does insulin do? Well, remember, it's a hormone. So it travels via your bloodstream. It's a chemical messenger. It has effects on multiple target organs. But the things you should know for GCSE, it causes glucose to be taken up by cells. Now, glucose is always taken up by cells because they need it for respiration. So I'm going to add on here, it causes more glucose to be taken up by your cells because we're trying to get it out of the blood, right? We're trying to lower your blood glucose. We want to get more of it out of the blood. So more glucose will be taken up by your cells because they basically become more permeable to glucose. But the most important one for you guys to remember is that glucose actually goes into your liver and into your muscles, and it is stored as glycogen. So glucose is a simple sugar. It's a monomer, but we're going to join them together and we're going to store it as glycogen, and that is in your liver and in your muscles. Now, because glucose is moving out of the blood into your cells, into your liver, into your muscles, it's getting stored away as insoluble glycogen, blood glucose levels will return to normal. They will fall back to the normal level. Now, higher tier only, you also need to know the other side of the story. So you need to know about a second hormone, which is glucagon. And glucagon is released from the pancreas when your blood glucose falls. So let's say you've not eaten for a long time, maybe overnight, maybe you've been fasting, maybe you've been exercising, so you've been doing a lot of respiration and you've been using up all your glucose to release more energy. Either way, glucose levels would drop. They would fall below normal. They've gone too low. So the pancreas is acting as the receptor. So the pancreas would detect the change. It would detect the stimulus. In this case, it would detect the fall in blood glucose and it would release glucagon. It would stop releasing insulin and it would release glucagon instead. Now, the way I remember this is glucagon. If you say it like that, it sounds like glucose has gone, right? So when the glucose levels are low, when the glucose has gone from the blood, the pancreas releases glucagon, okay? Now, what does this cause? This causes the liver, so the liver is one of the target organs, to convert glycogen back into glucose, and the glucose can then diffuse back into the blood. So remember those stores of glycogen that we stored in our liver, they get broken down back into glucose and the glucose diffuses back into the blood. Okay, and that is to bring our blood glucose levels back to normal or to increase them back to normal. Okay, quite a lot to learn for this topic, but make sure you've got your hormones right. So if blood glucose levels rises, the pancreas releases insulin. If blood glucose levels fall, the pancreas releases glucagon. Something else I will say here is these words are very, very similar looking, but they are very different things. So glucagon is a hormone. It's a chemical messenger released from the pancreas, yeah? When the blood sugar levels are too low or when the glucose is gone, the pancreas releases glucagon. Glycogen, on the other hand, is a store of glucose. So it's actually a carbohydrate. It's a store of glucose in liver and muscles. So insulin causes the formation of glycogen, whereas glucagon causes the breakdown of glycogen. So write those words down. They're very similar. You've got to spell them right or the examiner won't know which one you're talking about, and you've got to know they are different things. I've just written all over this. I'm going to have to rub it out so we can take you through a nice little summary diagram to put all of this together, because I know it is a pretty tricky topic. So let's look here. So this is normal blood glucose. We don't need to know the numbers, but it says you want about 90 milligrams of glucose in 100 milliliters of blood. That is your normal level. And that's what we want to maintain via homeostasis using negative feedback. We want to maintain that normal level. Now, if blood glucose levels rise after eating carbohydrates, the pancreas will detect this and the pancreas will release the hormone insulin. Let's follow it round. Insulin causes body cells to take up more glucose. It causes the liver to take up glucose and store it away as glycogen. It also causes the muscles to take up more glucose and store it away as glycogen. Now that brings blood glucose levels back down to normal. The other side of the story, which is higher tier only, if your blood glucose level drops below a set point, so maybe you've been exercising and using up a lot of glucose, that will be detected by the pancreas, but the pancreas will release the hormone glucagon because the glucose has gone from your blood, it's too low. Glucagon causes the liver to break down glycogen back into glucose. The glucose diffuses back into your blood to bring your blood glucose level back up to normal. Now, you could get a 6 marker, especially if you're higher tier, a 6 marker about control of blood glucose concentration, where you'd have to talk about insulin and glucagon, what they do, where they're released from, and the fact that this is an example of negative feedback to maintain the blood glucose concentration at a stable or normal level. Okay, linked to blood glucose regulation is diabetes. Now, diabetes is a condition where blood glucose levels cannot be properly controlled. But there are 2 different types of diabetes. We have type 1 and type 2. So we're going to go through a comparison of them side by side. So type 1 diabetes is caused because the patient's pancreas produces very little or no insulin. It is not working, it is not producing the hormone insulin. Whereas type 2, they are still producing insulin, their pancreas still produces insulin, but their body cells are not responding to insulin, or their body cells are not sensitive to insulin. Now, type 1 diabetes usually develops in childhood or very early adolescence, whereas type 2 usually develops later on in life in adults, and it is linked to certain risk factors such as obesity. Now, remember, a risk factor is anything that increases the likelihood of you developing a disease. It doesn't mean you're definitely going to get the disease, but it increases the chances that you will get the disease. Now, there are risk factors for type 2 diabetes. Now, obviously, obesity or being overweight is a big risk factor, but also a lack of exercise is a risk factor. Even age is a risk factor. As you get older, you are more likely to get type 2 diabetes. Family history as well could be a risk factor. Poor diet, so a diet that's very high in carbs, very high in sugars, obviously is a risk factor for type 2 diabetes. Now, if we compare the blood glucose levels with type 1 diabetes, because their pancreas is not producing insulin, their blood glucose levels become very, very high because there's no insulin to cause the glucose to move into the liver, move into the muscles and be stored as glycogen. Type 2 diabetes, again, it can become high because their body cells are not responding to the insulin the way they should be. Now, treatments, if you're type 1 diabetic, you will be regularly monitoring your blood glucose levels and you will be taking injections of insulin because your pancreas does not make insulin. So you need to inject it regularly, often before you eat a meal. Type 2, though, usually it can be controlled with a low carbohydrate diet and regular exercise. Now, really important that you don't just say balanced diet, healthy diet. That's not good enough. It's a low carbohydrate diet or a carbohydrate controlled diet, as well as regular exercise. Sometimes they do need medication. There are various examples of which you don't need to know, but like medication to reduce the absorption of glucose from the small intestine. There's one called metformin that you might have heard of. They might take insulin. Occasionally they do take injections of insulin to really boost their insulin, but it's not always worthwhile because remember, if you're type 2 diabetic, your pancreas is still producing insulin. It's just that your body cells are not responding to it or they're not sensitive to it. Now, there is no cure for type 1 diabetes. There's no cure really for type 2 diabetes. However, if you're early onset, so it's not fully developed yet, you can kind of reverse the effects of type 2 diabetes through lifestyle changes, such as adjusting your diet to be low carbohydrate diet, losing weight, and starting to take very regular exercise. Moving on now to hormones in human reproduction, which is a big topic in Module 5. So in males, not much to learn, but in males, you should know the hormone is testosterone. That is the main male reproductive hormone. Testosterone is produced by the testes or testicles, and it stimulates the production of sperm. That's kind of all we do for males. But in females, there are several hormones that are involved in the menstrual cycle. So let's go through them. We've got follicle stimulating hormone, which at GCSE, do not worry, you can just call it FSH. And FSH causes eggs or follicles to mature in the ovaries. Now, you should already know from what we've just done that follicle stimulating hormone, or FSH, is released from the pituitary gland in the brain. Okay, and it causes eggs or follicles to mature in the ovaries. Then we've got luteinizing hormone. Again, you can just call it LH, so don't stress. And LH, again, is released from the pituitary gland in the brain, and it stimulates the release of an egg from the ovaries, or it stimulates ovulation. Ovulation is the release of an egg. And then we have estrogen and progesterone, which are both released from the ovaries in females. They're involved in maintaining the uterus lining, so thickening and maintaining the lining of the uterus. Now, the menstrual cycle is a monthly cycle. It's about 28 days on average. It can vary slightly from female to female, but its role is to prepare the female body for pregnancy. And we're going to go through these hormones now in more detail. So FSH, you've got to learn this, right? FSH is a hormone. It's a chemical messenger, travels in the bloodstream. It's made by the pituitary gland. And in terms of functions, as we've said previously, it stimulates an egg to mature in the ovary, or you can call it a follicle. It also stimulates the release of the next hormone. So we're doing these in order, and I would recommend you talk about them in this order if you get an extended response question. So talk about FSH first. From the pituitary gland, stimulates an egg to mature in the ovary, and it stimulates the release of the next hormone, estrogen. Estrogen is a hormone that is released from the ovaries. It causes the lining of the uterus to thicken in preparation in case the egg is fertilized and it needs to implant. In terms of how it interacts with other hormones, it blocks the release of FSH, or it inhibits FSH, so that we don't get more than one egg maturing. But it stimulates the next hormone. So you can see FSH stimulates estrogen, estrogen stimulates LH. Now, LH is another hormone that's released from the pituitary gland, and this hormone is key because it triggers ovulation, or the release of the egg cell from the ovary around day 14, or in the middle of the menstrual cycle. The fourth hormone we have is progesterone. This is released from the ovaries after ovulation. After ovulation, what remains of the follicle is a structure called the corpus luteum. It's actually the corpus luteum in the ovaries that releases progesterone. But for GCSE, you can just say progesterone is released from the ovaries. It maintains the thick uterus lining, ready for implantation if the egg cell is fertilized by a sperm cell. It also inhibits both FSH and LH because you don't want multiple eggs maturing or multiple eggs being released from the ovaries. But obviously, if there's no pregnancy, if the egg is not fertilized and there's no implantation, progesterone levels will start to drop, the uterus lining will start to break down, and then we go back to the start of the cycle with menstruation or having a period. So really simple. FSH and LH are the two that are released from the pituitary gland. Estrogen and progesterone are the two hormones that are released from the ovaries. If you learn them in this order, it helps because FSH causes the egg to mature in the ovaries, and then it stimulates estrogen. Estrogen causes the uterus lining to thicken, and then it stimulates LH. LH causes ovulation, the release of the egg, and then progesterone maintains the uterus lining, keeps it nice and thick in case the egg is fertilized and needs to implant. Now let's look at it on a graph. So the menstrual cycle lasts for 28 days on average. Now the first part of the cycle, day 1 to 14, FSH is increasing gradually. You can see it here. It's only a slight increase, but FSH is increasing, and we know that FSH causes the egg to mature. We also know that FSH stimulates estrogen. So you can see estrogen is increasing as well. And estrogen causes the uterus lining to thicken. Then estrogen stimulates LH. So LH, which is this one here, has a big surge or a big rise, which triggers ovulation around the middle of the menstrual cycle. The egg is released from the ovary. That's what ovulation means. And that happens around about day 14. And then we have progesterone. Where's progesterone? Here. You can see progesterone levels are rising from day 14 to 28 to keep the uterus lining thick. But you can also see on this graph, if the egg is not fertilized, progesterone levels start to fall, which means the uterus lining will start to break down and menstruation will start to occur, or having a period. And then the cycle starts again, because when progesterone levels fall, FSH levels will then start to rise and another egg will start to mature in the ovaries. Now, if you're higher tier, it's a little bit more complicated for you because they do expect you to know how these hormones interact. So let's just go through that again. It's kind of like the knock-on effects of one hormone to the next hormone. So FSH, which remember is released from the pituitary gland, we said, I feel like I want to do it all again here. It's released from the pituitary gland and it stimulates an egg cell or a follicle to mature in the ovaries. But it also interacts with other hormones because it stimulates estrogen. So FH causes an egg cell to mature in the ovaries, but it also stimulates estrogen so that the uterus lining starts to thicken. Now, estrogen causes the uterus lining to thicken, but it also blocks any more secretion of FSH from the pituitary gland because we're trying to make sure only one egg matures. So we don't want more and more FSH to be released because then multiple eggs would mature in the ovaries. Estrogen stimulates LH, so LH levels start to rise. LH then obviously stimulates ovulation, which occurs in the middle of the cycle around day 14. Now, progesterone is the one that maintains the thick uterus lining, but it inhibits, blocks, stops the release of both FSH and LH. Why would we want to block FSH and LH? Well, we want to block FSH so multiple eggs do not mature in the ovaries. We also want to block LH so multiple eggs do not get released from the ovaries or multiple eggs are not ovulated, because if we did get ovulation of multiple eggs could be fertilized and that would lead to a multiple pregnancy, which obviously is more dangerous for the mother to carry multiple pregnancies and for the fetuses themselves developing and having to share nutrients and space, etc. It is more risky. It's more risky. They'll be born prematurely, low birth weight, etc. Okay, so if we go back now, I'm not going to go through it all again, but if we go back a second to this table, this is what I'm going to suggest you learn because it has the 4 hormones, it has where they're released from, and it has everything about what they do, whether you're combined or triple, combined foundation or combined higher even. It has everything they do and how they interact with each other. So FSH causes eggs to mature, but it also stimulates estrogen. Estrogen causes the uterus lining to thicken. It blocks FSH. It stimulates LH. LH causes ovulation, the release of an egg from the ovary. Progesterone maintains the thick uterus lining. It blocks both FSH and LH so that multiple eggs are not released from the ovaries. Okay, so I think we've covered everything. Again, this could be like a 6 marker where you name the hormones, where they're released from, what they do, and if you're higher tier, you would even have to include how they interact with each other. Let's now move on to contraception. So contraception is the use of methods or devices to prevent pregnancy. It works by either stopping the release of an egg from the ovaries, because if there's no egg cell for the sperm to fertilize. Or it could prevent the sperm from reaching the egg, because if the sperm can't reach the egg, can't fuse with the egg, fertilization cannot occur. Finally, it could work by stopping any fertilized eggs from implanting. So maybe the sperm does fuse with the egg, maybe fertilization does occur, but the fertilized egg cell or the zygote cannot implant into the uterus. Hormonal methods of contraception, we're going to look at the combined pill, the mini pill, the patch, there's the injection, there's the implant. There's also intrauterine devices or coils, which I haven't got in this presentation, but they can actually release the hormone progesterone. Releasing the hormone progesterone, obviously progesterone blocks both FSH and LH, so it would stop eggs maturing and it would prevent ovulation, so there'd be no egg cell for the sperm cell to fertilize. But let's go through these most important ones. And this could kind of come up on your exam as like an evaluate style question, like pros and cons. So the combined pill contains both estrogen and progesterone. Why? Well, estrogen inhibits FSH, progesterone inhibits FSH and LH. So basically no egg is going to mature, no eggs are going to be released from the ovaries. It is highly effective if it's used correctly. However, it must be taken daily, so you have to remember to take it every day. You might forget. And it does cause side effects like nausea, feeling sick, mood swings, that kind of thing. Now, the mini pill is very similar, but the mini pill only contains progesterone, not estrogen. But again, it works in the same way in that progesterone inhibits FSH and LH, so eggs do not mature and egg cells are not released from the ovaries. It also thickens cervical mucus, so it's very hard for any sperm cell to even reach the egg cell if an egg cell is released. It's good for women who can't take estrogen. It has fewer side effects than the combined pill. Because it is just progesterone only. So it does have fewer side effects, but just like the combined pill, it has to be taken every day. You might forget it, and if you forget to take it, then it's not as effective, right? It's not quite as effective as the combined pill, as it does not contain estrogen, but it does contain progesterone, so it should still block both FSH and LH, which means eggs do not mature and they are not released from the ovaries. The injection is an injection of progesterone. We know that progesterone will stop ovulation because it inhibits both FSH and LH, same as the pills. It also thickens cervical mucus, which makes it very difficult for sperm cells to reach any egg cell. It lasts longer, so 8 to 13 weeks. You don't have to remember the length, but it does last longer. No daily pill to remember. Again, there are side effects and there can be a delay in returning to fertility. So if you decided that you did want to get pregnant, you would have to wait a little while before your fertility returned to normal. The implant, which is implanted under the skin, again, uses progesterone. It releases progesterone slowly. Again, it prevents ovulation because progesterone inhibits FSH and LH, so eggs do not mature and there's no ovulation. Again, it thickens cervical mucus. It lasts even longer, up to 3 years. It's very effective. However, it does involve a minor surgical procedure to put the implant under the skin. It may be uncomfortable or slightly painful. Small risk of infection, you could say. And again, there'd be a delay in the return to fertility. The patch sticks to the skin. It releases estrogen and progesterone, which move through the layers of the skin into the bloodstream. Works like the combined pill because, again, estrogen blocks FSH, progesterone blocks FSH and LH, so eggs are not released from the ovaries. Really easy to use. You just stick it on the skin. You don't have to remember to take a pill every day, but obviously you do have to remember to replace it because it only lasts for about 7 days. It could cause skin irritation. And as I said, after the 7 days, you would have to remember to replace it or it would become ineffective. Now, you will quite likely get some information in your exam that you have to use to put together and evaluate, but I think it's really useful to have an idea of some of the pros and cons before going into the exam. So think about how effective they are, think about how long they last, think about side effects. With all of these, one of the cons you can say, which isn't on the slide but is really important, there is no protection against sexually transmitted infections or sexually transmitted diseases. So that is a con for every single one of these hormonal methods of contraception. Then we move on to our non-hormonal methods. So these don't contain hormones like progesterone or estrogen. We've got things like condoms, male or female condoms. They're what we call a barrier method. It's called a barrier method because they literally prevent the sperm from reaching the egg. If the sperm can't reach the egg, there's going to be no fertilization. They do protect against sexually transmitted infections. That's a big pro that you should be able to give. They are easy to use, however, not always effective because they might break, they might slip. If they're not used correctly, they are not going to be effective. Another barrier method, diaphragms, which go inside the female vagina and kind of sit over the cervix. And you can use diaphragms with spermicide as well. It's another barrier method because you're preventing the sperm from entering the uterus. So you're preventing the sperm from meeting and fusing with an egg cell. And if it's used with spermicide, it can make it extra effective because a spermicide is a chemical which kills sperm. Diaphragms are reusable, no side effects, relatively easy to use, right? And if used with spermicide, they can be very effective, but they are less effective than condoms. And again, they must be fitted correctly. Let's think about spermicides alone. I mean, we've already said it's a chemical that kills sperm. On its own, it's not very effective. And spermicides can cause irritation, but they're more effective if they're used alongside other barrier methods. So they're used alongside a diaphragm or a condom, for example. There's a type of coil, it's a non-hormonal type of coil, which basically sits inside the female uterus. The copper is toxic to sperm and eggs. It prevents fertilization, it prevents implantation. It can last for up to 10 years. And this is hormone free, so it has few side effects. It lasts a long time. However, it can cause heavier periods. There's no sexually transmitted infection protection. It needs to be removed and put in by a professional, can cause cramps. But the big pro there, I suppose, is the length of time that it lasts for. Then we've got two kind of other types of contraceptives, which you should be aware of. Abstinence is basically abstaining from sexual intercourse. Now, you could abstain from sexual intercourse full stop, or you could avoid intercourse during the fertile window. Now, the most fertile window would be around day 14 because that's when the mature egg cell is being released from the ovary. So you could avoid sexual intercourse around day 14 or day 10 to day 20. No side effects, it's free. However, it is very, very unreliable without careful tracking of the female menstrual cycle. Sterilization is a surgical method. It involves a surgical procedure. In men, you can cut the sperm ducts. In females, you can cut the oviducts. This is going to prevent the sperm and the egg from meeting, so there's no fertilization. It is permanent. It lasts forever. Very unlikely that you could reverse it. No daily effort, nothing to remember. But then that's also a con, right? Because it's irreversible, you might change your mind. You'd have to be very certain that you wanted to do this. You did not want pregnancy. It involves surgery, so there are risks of anesthetic, risks of infection. You can always talk about those risks with any surgical procedure. Now, higher tier only, as well as looking at contraception, you also need to look at how we can use these hormones to treat infertility. Now, infertility is when a woman finds it very difficult to become pregnant and wants to try and become pregnant. So it's kind of the opposite of contraception, right? Now, doctors can give women FSH and LH as fertility drugs. So they can give them injections of FSH and LH. Why? Well, now we've done our menstrual cycle hormones, you should know why. They'd be given FSH because FSH stimulates the eggs to mature. So maybe her egg cells are not maturing. Maybe there's an issue with the amount of FSH her pituitary gland is producing. So by giving them FSH, it's going to stimulate egg cells to mature in the ovaries. By giving them LH, it's going to trigger ovulation or the release of an egg cell from the ovaries. And this is obviously going to increase the chance of pregnancy because there will be mature egg cells. They will be released from the ovaries. They will be present in the oviducts, ready for fertilization by a sperm cell. Now, obviously, this is going to help women who might not otherwise be able to conceive. It's less invasive than IVF, which we're going to look at in a second, but it doesn't always work. And it does increase the chance of multiple pregnancies or multiple births. So it's more likely the woman would have twins or triplets if she did become pregnant. That is more dangerous for both the mother and the baby. The baby might be born with low birth weight, born prematurely. The labour is going to be more complicated. So it does increase the risks. And having injections of these hormones, FSH and LH, can cause side effects like mood swings, headaches, nausea, and it can cause emotional stress, right? If this does not work, it can be emotionally very stressful. Let's look at IVF. This again is higher tier only. This is a fertility treatment where fertilization happens outside the body. It stands for in vitro fertilization, which means fertilization or the fusion of the egg and the sperm cell is occurring outside of the body in a test tube, in a petri dish, in the laboratory. So let's go through the steps. So the women would first be given injections of the hormones FSH and LH because this is obviously going to stimulate egg production, make sure that the egg cells are fully matured and ready for ovulation. Then there is a surgical procedure to collect several eggs from the ovaries. The eggs are then mixed with sperm in a lab. This is why we call it in vitro fertilization, because the sperm cells will fertilize the egg cells in the test tube or in the petri dish. Now, those will be monitored to see if fertilization has taken place. And if fertilization has taken place, then we'll start to see embryos develop by mitosis, because the sperm cell fuses with the egg cell to form the fertilized egg cell or the zygote. And then that zygote will start to divide by mitosis. And you'll end up with a cluster of cells that we call an embryo. Those embryos will be studied to check that they are growing as they should be, that they're dividing successfully. And then 1 or 2 embryos are placed back into the woman's uterus or womb. Obviously, placing 2 increases the chances of success, but it does also increase the risk of multiple pregnancies. If the embryo successfully implants, then it could lead to a successful pregnancy. Okay, so you do have to learn these steps if you're higher tier. Now, pros, it helps couples who cannot conceive naturally to have a baby. It increases their chances of a successful pregnancy. You could do this with donor eggs or donor sperm if needed. So if either the male or the female had a genetic disease, you could use donor eggs or sperms. Cons, right, it's an expensive procedure. It's a very expensive procedure. Low success rates, that's really important to remember, especially in older women. So women over the age of 40, the success rates are very, very low. Physically and emotionally stressful because it does involve hormone injections for the woman, a surgical procedure, to remove the eggs. And if it doesn't work, and there's a high chance it won't work because it's got low success rates, that's very emotionally stressful as well. Can lead to multiple births, which are a risk to both the mother and the baby. And there are ethical concerns because embryos might be created that are not then used or implanted. And those embryos potentially could be frozen and used for research, but they might be destroyed. Now, don't just say it's unethical. You'd say some people have ethical concerns because embryos may be destroyed. An embryo is a potential life which could be destroyed if it's not used in the IVF process. Now, embryos can be screened or have their DNA studied to detect inherited disorders before they are implanted into a mother's uterus. This again is higher tier only. This is embryo screening. So this might be one reason why a couple opts for IVF, because they don't want to conceive naturally. They want to create the embryo in the lab so the embryo can be screened or checked for genetic diseases before it is implanted. And this would be if the couple knew that there was a family history of a genetic disorder, or maybe they were both carrying a recessive allele for cystic fibrosis and they were worried that their child could potentially inherit those alleles and inherit that genetic disorder. So embryo screening involves actually studying the DNA of the embryo as it is developing in the lab. Now, some people argue that embryo screening should not be allowed. And again, we're going to talk about like the cons, if you like. When an embryo's DNA is studied, the embryo might actually be damaged in the process. It's a stressful process for the mother and father, but ultimately if they opt for it, they give consent, they obviously want to find out, but they might find out that their embryos have inherited a genetic disorder. The screening process is very expensive. The embryo itself, which you can say has a right to life, cannot give consent to the screening process and it might be damaged in the process. You could say it demonstrates an increased prejudice against people with inherited disorders, because it's kind of like you're saying, well, if that embryo has an inherited disorder, we're not going to implant it. You're making a decision on the right to life of that embryo, which could lead to prejudice against people that are living with inherited disorders. Right, a couple of topics left. Adrenaline, higher tier only. Another hormone that you need to learn about. So we're moving away from the reproductive hormones now. Adrenaline is a hormone produced by the adrenal glands. Remember, you have two adrenal glands. They sit on top of your kidneys. It's released in response to fear or stress. It prepares the body for fight or flight. We call it the fight or flight hormone. Remember, it's a hormone, so it travels in the bloodstream. It's released by a gland, the adrenal gland. It's a chemical messenger. But it does have widespread effects because it has multiple target organs. So things you should know: it will lead to increased heart rate and increased breathing rate. Why? Well, this is the fight or flight hormone, right? It's preparing your body to run or fight because you're in a fearful or stressful situation. So if you increase your heart rate and breathing rate, it's going to increase your intake of oxygen and the delivery or transport of oxygen to your cells. Increasing your heart rate not only means that oxygen will be delivered faster to cells, but glucose will also be delivered faster to cells, specifically like your muscles or your brain. Because if you're in a fearful situation, you need to think clearly about how to respond, and your muscles might need to do lots of contraction. So they're going to need to do lots of respiration to release that energy. So they're going to need lots of oxygen and glucose. So breathing rate increases to take in more oxygen, heart rate increases to boost the circulation of blood to deliver more oxygen and glucose to your cells. Now, because your heart is beating faster and blood flow is increased, it does increase blood pressure. Other effects causes your pupils to widen, which again makes sense because you need to be more alert. You need to be able to see clearly what's going on if you're in a fearful situation. It also leads to the breakdown of glycogen into glucose. Again, all these things should make sense. If you need to release more energy and you need to do more respiration, you're going to need more glucose. So your stores of glucose, which is your insoluble glycogen, will be broken down into glucose, like in your muscles, be broken down into glucose. So you have more glucose can do more respiration, release more energy for muscle contraction so you can run away. Now, adrenaline is not controlled by negative feedback. It's not the same as blood glucose regulation with insulin and glucagon. It's a short-term emergency hormone. So it is released from your adrenal glands when you are scared or stressed, when your body thinks you are gonna need to run or fight, and you need this increase in heart rate and breathing rate and an increase in the rate of respiration. Thyroxine is the last hormone we're going to do. Again, this is higher tier only. Thyroxine is released from your thyroid gland, which is here. It controls your basal metabolic rate, which just means your base or your normal metabolic rate. Now, metabolic rate is the rate that chemical reactions happen in your body or in your cells at rest. So it's the speed of chemical reactions, which you should know from module 4 when you do metabolism. It's the speed of chemical reactions in your body or in your cells, but thyroxine is the hormone that controls your metabolic rate. Now, thyroxine levels are controlled by negative feedback, so we're going to have a little look at that. So if thyroxine levels fall, where are we? Here's your thyroid gland. You've got a fall in thyroxine levels. Okay, so we're on this side. So if your thyroxine levels fall, this is acting as the stimulus, right? There's a drop in thyroxine levels in your blood. The pituitary gland, which is known as the master gland in the brain, will release more thyroid stimulating hormone, or TSH. Thyroid stimulating hormone will travel to the thyroid gland and stimulate the thyroid gland to release more thyroxine. So your thyroxine levels return to normal. Now you can see this is negative feedback, right? Because a drop in thyroxine levels initiates a response to reverse the change or to increase the thyroxine levels. So if thyroxine levels fall, that might mean your metabolic rate drops. The pituitary gland releases more thyroid stimulating hormone. That causes the thyroid gland to release more thyroxine to return your thyroxine levels to normal. That's why it's called thyroid stimulating hormone, right? From the pituitary gland, because it stimulates the thyroid to release more thyroxine. That means your thyroxine levels will increase. That means your metabolic rate will increase. Your rate of respiration will increase back up to normal. Now, the other side of this, have I done the other side? Yes. So if you have a rise in thyroxine levels, now this means levels of thyroxine are increasing, your metabolic rate is increasing too much, your rate of respiration and other chemical reactions is increasing too much, the pituitary gland would release less thyroid stimulating hormone. So you'd have less of it going to the thyroid gland. The thyroid gland would then release less thyroxine to bring your thyroxine levels back to normal. Your metabolic rate would then drop back down to normal or become stable again, and your rate of respiration would go back to normal and become stable again. So quite a lot to remember on this slide, but thyroxine from the thyroid gland controls your metabolic rate. But thyroxine is controlled by negative feedback, and that involves another hormone, thyroid stimulating hormone. So if thyroxine levels fall, more thyroid stimulating hormone from the pituitary gland leads to an increase in the release of thyroxine from the thyroid gland. But if your thyroxine levels rise, you'll have a reduced release of TSH from the pituitary gland. So your thyroid gland will start releasing less thyroxine. So your metabolic rate will decrease back to normal. And that is it for this video, because this has covered all of the content applicable to both combined and triple in module 5. Now, I'm warning you, if you're triple science, there is a huge amount of triple only content in module 5. It's like double the amount for you guys. So I'm going to be posting a second video with the triple science only content from Module 5, including the brain, the eye, temperature regulation, etc., the kidney. So make sure you're subscribing, turning on your notifications. So if you're triple science, you can get that video because you really, really need to watch it. For everyone that's watched this video, I hope it's been useful. And yeah, let me know in the comments how you found it. I hope you found that video useful. Make sure that you are subscribed to this channel and that you've switched on your notifications, because very shortly we're going to be posting a second part to this video, which is for all you triple science students, because there's loads more triple content that you guys need to cover for Module 5, such as the kidney, the eye, and temperature regulation, as well as tropisms. Make sure you've clicked the link in the description to access your free flashcards, and click that link to go to my resources for all of your biology GCSE revision needs. I'll see you shortly for the next video.